Assalamu alaikum and welcome to our channel. I am Dr. Hamad Ismail. Today we will talk about the peptide docking, or we can also say it protein peptide docking. Well, why we do protein peptide docking? Protein docking is sometimes not feasible because the protein sizes are so large, so it took large of time to complete it. So the short way is protein peptide docking. What we can do in protein peptide docking, we know the active site of our one protein and we want to dock it with our other protein, or we know that from one protein there is a specific fragment of interest or specific amino acid or specific 2-3 amino acid which may be your point of interest to dock with the protein. So that's why instead of running the whole protein docking, which took a lot of time, we prefer to perform the peptide protein docking. Today we will demonstrate that how we can perform peptide protein docking in Schrödinger. So let's start. Open the Schrödinger program. In this tutorial, we are using Schrödinger 2024. Go to File. First of all, change the working directory. On the desktop, I have created a folder with the name Protein Docking Demo, Peptide Docking Demo. Select that folder. Then go to File again, create a new project. In that folder, I am going to create a new folder with the name Demo and save it. Now, go to File again. You can get the PDB or you can import the structure. For this demo, I have selected the protein 1GYB. 1GYB is a multi-chain protein, that's why we choose it. Select it and open it. Import the structure and it is done. So you can also get PDB and type 1GYB and it will be directly downloaded, or you can download it from the protein PDB structure. So this is our protein. This is our protein of interest. This is a relatively big protein with multiple chain. This is the sequence of protein. If you are unable to see this point, you can click on this plus sign and here you can see a sequence viewer. When you click on the sequence viewer, it will show you the complete sequence of the protein. These are the multi-chain protein. You can see protein chain A, protein chain B, and these are their sequence. Here you can see there are also some protein with some short amino acids, like chain A is consisting of some protein, E is consisting of some small protein, F is some other small protein, and so on. So for now, I let it minimize. Here is our working directory and here is our complete protein. If you click on this protein, it will be highlight. This one, these are the chains. Protein chain A, protein chain B. You can see these proteins are highlighting here. Protein chain C, protein chain D, and protein chain E. If I double click on it, it will highlight the protein chain E. In this tutorial, I will separate this protein chain E. I simply right click, select it and right click it on it. And I say copy to the new entry, or you can extract to the new entry. Because I want to see the interaction, I just simply click on the extract to the new entry. Extract to the new entry means it will extract protein from here, delete from the original source and make it a separate entry. It is asking me to change the name. I change it as a chain E and enter. So the name chain E is chain. If you see over here, this is a single chain protein. But if you click on this protein and go over down here, you can see protein chain E is no longer part of the main protein. If you want the main protein as the same, you can copy it or you can duplicate it. The other way is doing it. You can simply copy paste it or you can extract it. This is for the demo. If you have your own protein, just simply go to the import structure and import your protein as you like. If you see in the structure of protein chain E, it is consists of 5 amino acids. Amino acid G, F, S, F and G. So I want to see the effect of or interaction of these 5 amino acids, this small peptide with this complete amino acid chain. I want to see either this chain interact with this protein or not. So the first step is the concept and the extraction of the protein. Now the second step is we need to prepare the both protein. We need to prepare the protein as well as the chain E. The first step is the protein preparation. If you cannot find the protein preparation wizard here, you can go to the task and search it here, protein preparation wizard. You can find the protein preparation workflow. Here is the protein preparation workflow. Normally it is present over here. I click on the protein preparation wizard. It shows the following window. If the protein preparation wizard contains specific protein, pre-processing, optimize hydrogen bond, optimize or delete the water molecule, I just change the name to 1GBY and keeping the default setting, I click on the run button. If you want to see the setting, it is using the 4 processor. I want to speed it up. I increase the number of processor to 8. It will prepare the protein quickly and click on the run. Now it is submitting the job. You can see here the option is saying that it is submitting the job for the protein preparation. Now you can see that the job is started and it is counting. Now you can close it. If you want to see the progress over here, the job portal is green and it is protein preparation 1 is running. If you want to monitor it, you can go over here and this is the running job. You can double click it here and it will give you the details of the running job and so on. It will take few minutes to complete the job according to your computer speed. Our protein has been prepared. This is the prepared protein, you can see. Now the second step is chain E protein preparation. In the same way, go to the protein preparation. I change it the name as chain E and click on the run button. It will submitting the job and start the job. Close this window and it will complete the job and prepare the protein. Chain E has been also produced. Now we are going, our both chains are produced. Now we are going to perform the peptide docking. For peptide docking, go to task, browse, and in browse there is biologicals. In biological we have peptide docking. Under docking session we have peptide docking. Click on the peptide docking, this window will appear. Here there are two parts. This is the grid generation and second part is the peptide region. Here I have selected the protein chain E and I am going to nominate because this is 5 amino acid protein. This is a small peptide. I am going to choose this one. I select it here. After selecting, click on the project table. When this molecule is selected and you will click on the project table, this peptide will appear here. Now minimize it. After minimize it, select this one and this one. Now the main chain is selected. Go to the peptide menu again and click on the centroid for selected residue. Do not choose the existing grid because it generates the problem. The best possible option is click on the centroid for selected residue, select the residues. After residue, click on all. It will include all the, you can see all the residues have been selected, and click all and then click OK. When you will click OK, it will generate a grid around it. Go back to the window, then allow cis-amide bond, it is your choice. You can return the number of poses as you like. For demo, I choose it as 5. You can choose the scoring method MMGBSA or glide score. Both methods can be used. Either you can use MMGBSA or glide score. Glide score is a faster method and MMGBSA will take some more time, which means you can perform peptide docking on the basis of glide score as well as which is the docking score, or you can also run on the MGBSA. Here is the docking. I will choose as the GS score, that's it, and go to the wheel cycle. Here I choose the maximum number of processor so that it run the job quickly. If you reduce the number of processors, the job will take more time. If you make the higher number of docking poses, it will also make more time. When you have completed the setting, click on the run button. Now it is submitting your job. You can see the job is now starting. The wheel is now running. Close this one. Your job is now running. You can see the job status over here. Here peptide GS, you can see into this one, and this one, this is the docking GS running, and you can see the status that job 4 of 1 is running. It will take few minutes to complete the job depending upon your computer speed, depending upon the number of poses you choose, depending upon you have choose MMGBSA or glide score. When the job will complete, the result will appear over here. Peptide docking result has been produced. You can see when the results are produced over here. You can see these. I should drag it over here. These are the peptide docking GSP output. The first one is our receptor. If you click on this one, this is our receptor, and these are basically our ligands. Okay. Now what you need to do, you need to, on this circle, you need to double click on it. When you double click on it, it will fix. Then the ligand will be added. If you look into this area, when I click over here, the ligand appear. Again, kindly look into this area. When you click on this, the ligand is appearing. And these are basically different poses of peptide which dock with it. You can select the both one to see the interaction. To get the better visualization, you can select this one, and you can go to preset and you can choose the bioluminosity default. When you choose the bioluminosity default, it will give you the this type of colorful structure. Let me hide this one to make more space. Okay, you can see these are our structure, and these are our over here, these are our ligands. If you press the Alt button and right click on the mouse and rotate, it will rotate. Okay, these are, you can see this is our ligand. If you select the ligand in this one, you select this ligand, if you click on the E, it will focus. You can see this is the focus one. The complete ligand is selected. What I did, I came to this part. This is the protein and this is our peptide. I opened it. After opening it, I selected the E chain and I double clicked on it, and it is automatically zoomed in, and this is the molecule which is completely selected. Now I am going to style, and in the style I am going to give it some structure. It will say now it is bold, which means it is more visible. Now select the Alt button and press the left click of the mouse, and it will show you how these molecules are showing. You can zoom in or zoom out that how this molecule is present. If you want to change the presentation, you can go to the style and choose different kind of features you want to use. If you want to give it a specific color, like if I give it a pink color, the complete molecule is become pink. Or you can choose to give it the yellow color. From here I give it to the yellow color for better presentation. So this is my complete ligand. You can save this image, go to the file, and you can go to the export structure, and you can save it as a PDB format, as a mol file, or any other format you need to do it. Or select it, press the shift button and then select it. After that, right click on it and go to the export. Kindly have a look on it, select it, then press the shift button and select this pose. Both are blue highlighted. Then any one of on it, right click and you will see the export button. Before exporting it, when you have done this one and this one are selected, right click on it and you will see the merge button. When you merge it, you will see a new entry will appear. Now remove the double click over here and this one is now fixed with the changing you have just made. Now right click on it and go to the export button and go to the structure. From the structure, you can save this structure as a PDB format. It will save your protein or structure in a single PDB format in a location where you will save. The second option you can do is export and export to the spreadsheet. And I'm going to change the format to Excel and change it like I say demo pep and save it. Now it's asking, are you going to save the displayed one or the full one? If you save the full one, all the ligand and all the interaction of peptide will be saved. If you want to include the 2D image, it will also save the 2D image. Well, for demo, I save it as the full as the include image and go to export. It is now exporting. Depending on the structure, it will take some time to complete the import. Now I go to minimize the button. This one is my peptide docking demo folder. Here I have saved the file with the name demo pep. This one is the Excel sheet. If you open the Excel sheet, you will see all the information. Like here, this is the 2D structure. This is the protein information, and these the E chain. From this onward, these are the different peptide structures. On the top menu, you will see the multiple parameters present in it. You can study the parameter you like. What parameters are these? Let's have a look on Mastro. Here, go to the table. In the table menu, you can see this is the peptide docking and this is the protein one, and this is our prepared one, and this is our different poses of ligand. If you can scroll, here is the information we need. You can hide all the families. Now nothing is shown. What we are interested in the glide primary. Glide primary show you the glide score. The highest score mean -6. This is the highest score, which means this is the best score we got. If the value in the negative is higher, it means it is a good one. As you saw, the value is going to decrease as we go in the bottom, which means these poses are not so much useful. Normally, first 2-3 poses are of our interest. This way, this is the docking score, this is the glide score. If you want the more, you can see the all glide parameters. You can see all macro model scores, which include multiple properties like G score, ligand binding efficiency, lipo score, metal glide metal score, glide reward score, RMSD value, potential energy, and so on. All these information is already present on the Excel sheet we have just generated. If you look into the Excel sheet and you go on the left, you can see all the information are already downloaded. Like these are the grid center, grid range, glide score, docking score. If you look into downward these are the docking scores and so on. All the information are already present in this Excel sheet. You can extract your information of interest and you can work on it. So this table gives you the complete summary. So basically the glide score gives you the idea that how a particular ligand is going to interact with that particular protein. And you can make a different kind of poses and information of your interest. In peptide docking, we can also perform some other analysis. So let's have a look on them. If we go to the task and go to the browse, in biologicals, we have some option. First option is the structural interaction fingerprinting. We click on that, like if we click on this one and then come back to our window and click on the generate fingerprint. It will generate fingerprint for any contact including backbone interaction, side chain interaction, polar residue, hydrophobic interaction, aromatic residue, and so on. Click on the generate fingerprint. So it is saying you must need to select at least 2 entries. So we first select the protein, and the second entry we select is randomly a residue. Then we come back and then again click on generate fingerprint, and it is done. Display interaction. Now click on the display interaction matrix. It is showing you the interaction diagram. Like this is the any interaction. If you go on them and click on the hydrogen bond acceptor, so it is showing you the hydrogen bond acceptor. This 2D structure shows that hydrogen bond interaction is with that, which means this protein is showing hydrogen bond interaction, with this residue at position number. You can see the molecule in the left corner over here, aspartate B18. Ligand is showing GFSF, peptide is showing interaction with ASN B18. Similarly, this is another one. It is showing D11 of ASN. In this way, you can also see some polar residues. These are the polar residues. Then the hydrogen bond donors. This is the one single hydrogen bond donor. Then you can also see the aromatic residues and so on. If you like, you can save these images to add in your transcript. You can also add different the color main plant, and you can make some more interaction on the basis of glide score, E-sol, glide energy, E-model, and so on. If you like, you can export this file. All the data will be stored as the form of CVS, mean Excel sheet, like interaction fingerprint diagram, IFD. I save into the diagram and the file is exported. Let me explain you again. First, we have selected this combined form in which protein and peptide are merged. Then we came into the biologicals and biological into protein interaction analysis. Here we added the chain, all the chain except the E chain. We placed them in the one box and the protein E in the second box. We clicked on the calculate button, and it is saying that at D position protein chain E is interacting with amino acid phenylalanine at 43 position. So you will get all the protein. If you click on this one, it is also at chain B with the 63 position at serine 64 position. Similarly, all the interaction can be quantified in this way. You can find out on the basis of residues, on the basis, or you can choose the custom chains and so on. But the best way is using the chain. Then, and here is another option, export to residue scanning. If you click on this one, a new window will open. It says that it has stability. You need to bind the chain. All the chain are calculating, then residue and alanine, and you will apply. If you want to add mutation, you can add mutation, and then run the analysis. If there is any analysis present over here, it will produce result for you. It's just a quick overview of this method. You can explore these options in details. The next analysis you can do is go to browse, and in the protein analysis, in biologicals, in the protein analysis, we have reactive residue options. Here we want to see the reactive residues. I have selected this merged protein, in which protein as well as the peptide is present, and analyze workplace. I click on the analyze workplace, so it is analyzing all these molecules present on this interaction, and it is giving you the answers. Here it is D123 tyrosine. It has oxidation state, and this is showing the position. When you click on any position, it will show you the back that this is the position. This is deionization site with solvent accessible surface area, exposure and beta factors, and so on. You can also select proteolysis, aspartame, free cysteine site, oxidation site, deamination site, and so on. This is the other information which can protein-peptide docking provide. The next analysis you can perform, again go to browse and biological. Here is another option known as residue analysis. I have selected the same protein and run the residue analysis. It will take some time and gives you the idea about the residue analysis, about hydropathicity, potential energy, internal energy, interaction energy, solvent accessible surface area, and other parameters. So you can see the result has been generated. These are the residues, and these are the information about hydrogen atom. This is the residue number, and phenyl at 45 position, hydrophobicity. If you click on any residue, it will show you in background by highlighting the residue information at the background and the information. You can save the plot as save the figure and the plot, or the specific surface, like you can save this pose showing the interaction, and this information can be very helpful to reporting in your data. The last thing I want to explain here is the protein surface analyzer. If you click on the protein surface analyzer, if you select this molecule and click on the analyze button, it will analyze all the surface area according to their positive, negative charge, hydrophobicity, and so on. It will generate result in few minutes. These results are generated for the protein 1-GBY and peptide GFSF, which we selected. These are the detail, the positive. These are the basically the protein. Let me fix it. If you click on this icon, it will back to the full zoom, and these are the results. The blue color show the positive charge, Similarly, the red color show negative charge, green color show hydrophobicity, and so on. So these are the basically color patches, or you can also choose the scoring type. It will show you the score, it will not show the exact color. The negative and hydrophobic, only the selected patch if you want to see. It will give you the complete picture of the molecule. Here, if you can see, when we run the analysis, the S sign is appeared. If you right click on it, it will show the protein patches and hide the protein patches. You can show or hide the protein patches. Similarly, the other analysis, you can notice there are 3 lines over here. If you click on this line, there are some analysis like interaction analysis, finger analysis, and spectral clustering. These options are all also available for your view. This was the brief video about the peptide docking and peptide docking and the related analysis. You can explore the more option. If you have any queries, you can ask in the comment section. If you like our video, please like and subscribe. In the future, we will upload more video on Schrödinger. Thank you so much for watching. Allah Hafiz.